This is our third and final video on microbial metabolism. In the last video, we went over some of the key concepts of aerobic respiration, the process in which organisms completely oxidize glucose and, in turn, reduce oxygen. In this video, we are going to discuss some of the alternatives to aerobic respiration. We are going to start with anaerobic respiration and fermentation. We will also briefly discuss how other molecules besides glucose can be used as an energy source and some of the links between catabolic pathways and anabolic pathways. There are three main pathways for microorganisms to obtain energy from a molecule of glucose: aerobic respiration, which requires oxygen, anaerobic respiration, and fermentation, which do not. Organisms can obtain the most energy in the form of ATP from aerobic respiration. But what if oxygen is not available? Or what if an organism doesn't have the genes encoding the proteins necessary for aerobic respiration? This is where anaerobic respiration and fermentation come in. These additional pathways allow organisms to obtain energy from glucose, even if the environmental conditions or their genetic characteristics don't allow for aerobic respiration. Before we talk about anaerobic respiration and fermentation, let's quickly review aerobic respiration. Remember, aerobic respiration is the complete oxidation of glucose coupled to the reduction of molecular oxygen. Glucose oxidation occurs in three stages: glycolysis, the intermediate step, and the Krebs cycle. A little bit of ATP is generated during glycolysis and the Krebs cycle through substrate level phosphorylation. Most of the energy extracted from glucose is found in the high energy electrons carried by NADH and FADH2. These electron carriers bring the electrons to the electron transport chain. Electrons from NADH and FADH2 move through the electron transport chain to the final electron acceptor, oxygen. The energy from these electrons is used to build a hydrogen ion gradient that in turn is used to make ATP through oxidative phosphorylation. Okay, so that's aerobic respiration. Some microorganisms have the ability to use other inorganic electron acceptors besides oxygen. Some examples are nitrate, sulfate, and carbonate. In anaerobic respiration, microorganisms couple the oxidation of glucose with the reduction of these other electron acceptors. They will go through the process of glycolysis, the intermediate step, and the Krebs cycle, and using an electron transport chain, they will transfer electrons to these alternate electron acceptors. There's a lot of variety in anaerobic respiration, but it can play a very important ecological role in the recycling of elements throughout the environment. One example of this is in nitrate reduction. In nitrate reduction, organisms reduce nitrate to nitrite. They give electrons to nitrate to form nitrite. Nitrite can be released as waste, or it can be further metabolized to ammonium or to nitrogen gas. In the case of ammonium, microorganisms can use it as a source of nitrogen for building amino acids and other nitrogen-containing molecules. This is called the assimilatory nitrate reduction pathway. In other organisms, they may reduce nitrite to nitrogen gas, and that effectively removes nitrogen from the environment. Usually we're talking about soils, and returns that nitrogen back to the atmosphere. This is a process called denitrification. In the laboratory, we can characterize microorganisms based on their ability to reduce nitrate. And there's a test, it's called the nitrate test, where we grow the organisms in a medium that contains an excess of nitrate. We then can detect nitrate reduction by looking for the presence of nitrite. We add chemicals that react with nitrite to produce the red color that you see in this picture here. All right. In this test tube on this picture, you also have something called a Durham tube. That Durham tube is a little inverted miniature test tube that can capture gas. So let's say the organism reduces nitrate further to nitrogen gas. We can look for the evidence of that inside that Durham tube. So this is just another example of a way that we can characterize microorganisms using our understanding of biochemical pathways. Okay, so we talked about anaerobic respiration. Now let's take a look at fermentation. Fermentation is the partial oxidation of glucose coupled to the reduction of organic molecules. Partial is important, right? We don't go all the way through to carbon dioxide. We don't break that glucose molecule down completely to carbon dioxide. We only partially break it down. So glucose oxidation produces those reduced electron carriers like NADH. Remember my dump truck analogy? Those electron carriers must have a way to get rid of those electrons so that oxidation of glucose can continue. This is where fermentation comes in. Fermentation allows for the regeneration of NAD. So we have the partial oxidation of glucose coupled with the reduction of organic molecules. Right? And there's a variety of different organic molecules that can be used here. Pyruvate is the most common, and the products of fermentation include things like lactic acid, acetic acid, carbon dioxide, and ethanol. These are all waste products. The cell doesn't use those any further. So glucose, remember, glucose oxidation goes all the way through glycolysis, the intermediate step, and the Krebs cycle. But in fermentation, we don't use the intermediate step and we don't use the Krebs cycle. This is because these stages produce lots of electrons that have to be carried away by NADH and FADH2. Fermentations can regenerate NAD, but they're not very efficient and produce a lot of cellular waste products. Because of this, cells using fermentation stop glucose oxidation after glycolysis. So you have a little bit of NADH that is produced in glycolysis, and that NADH is used in our fermentation pathways. Those electrons are dumped in the fermentation pathways. Again, the purpose of fermentation is to regenerate NAD. So let's just look at a couple of examples of fermentation. One example is our lactobacilli. Lactobacilli are the bacteria that we find in our yogurt. These organisms obtain ATP through glycolysis, and then they regenerate NAD by dumping electrons back on pyruvate. So that's what you see in the second half of this pathway here. Lactic acid is produced as a waste product. And when these organisms are grown in milk, the production of lactic acid lowers the pH, and that causes the proteins in the milk to start to coagulate, to change shape and kind of clump together. And that's what creates the thicker consistency that we see with yogurt. Again, the purpose is to regenerate that NAD, right? That can go back up and allow for additional oxidation of glucose and additional production of ATP. Another example of fermentation in action is the production of alcohol by yeast. Yeast grown in grape juice produce wine, right? Again, these yeast are obtaining ATP through glycolysis. They're breaking down the sugar that's in the grape juice, and then they regenerate NAD through a different set of reactions than what we see in lactic acid fermentation. They regenerate NAD through these reactions that make ethanol and carbon dioxide. So here we have glycolysis followed by the NADH regeneration, that what we call ethanol fermentation process or alcohol fermentation process. These reactions are usually named by their products, and we are regenerating NAD. These are just two examples of human uses for fermentation, but there are lots of them, right? And a lot of it is used in production of foods. So kombucha, bread, beer, a variety of cheeses all rely on fermentation pathways, and the different products of fermentation provide the unique flavors in those food products. So there are lots of different fermentation pathways. They're all the same in the sense that their purpose is to regenerate NAD, and each is different in terms of the product that is made or the waste that is made. So again, fermentation itself does not make ATP directly. ATP is made through glycolysis, usually, and the purpose of fermentation is really just to eliminate electrons and regenerate NAD. There are too many to go into deep detail here, but many of our laboratory tests examine a microorganism's ability to perform fermentation. Most of these tests rely on growth media containing pH indicators, which change color in response to the change in pH. These would be examples of differential media, right? Because there's a difference in appearance based on growth characteristics of the organism. In the test tube you see on the screen, the pH indicator is red under neutral conditions, but yellow under acidic conditions. This particular organism is a strong fermenter. It produces lots of acid in its fermentation pathways. Because acids are a common fermentation end product, this is an easy way for us to look for evidence of fermentation. This particular assay also includes a Durham tube. This is like the one that we saw in the nitrate test. And these Durham tubes, all they do is they capture gases, right? So in the nitrate test, it was capturing the nitrogen gas that might be released as a product of nitrogen reduction. Here, it might be capturing carbon dioxide or hydrogen gas that are also common products of fermentation. So again, I'm not going to go into details of the different laboratory tests that look at fermentation pathways, but this is just an example of one of those tests. So far, everything we've discussed here has centered on glucose catabolism. Microorganisms are capable of extracting energy from lots of different types of organic molecules, not just glucose. Briefly, and very briefly, I want to discuss how catabolism of other molecules like lipids and proteins intersects with the pathways of glucose catabolism. Let's start by looking at lipid catabolism. In our lecture on microbial nutrition, we talked about exoenzymes. Remember, those are enzymes that are secreted into the extracellular environment to break down large biological molecules so that their components can be used as a source of nutrition. One type of exoenzyme is a lipase. Lipases break down lipids like triglycerides and phospholipids. These molecules are composed of a glycerol molecule with several fatty acid molecules attached. So here's an example of a triglyceride. The lipase enzymes break the bonds between the glycerol molecule and the fatty acids. Once they're broken down into these smaller components, they can be further catabolized by the cell. So glycerol can be oxidized into one of the intermediate molecules of the glycolytic pathway. So then it can feed directly into glycolysis. Fatty acids are oxidized in a process called beta oxidation, and the product of beta oxidation is acetyl-CoA, which is something that can immediately enter into the Krebs cycle. Likewise, proteins can be broken down into individual amino acids by proteins called proteases, and we looked at those when we were looking at our exoenzymes. Remember, all amino acids have the same core structure with an amino group on one side, a carboxyl group on the other side, and a variable group, or R group, in the middle. Microorganisms can use enzymes to remove the amino group in what's called a deamination reaction, or they can remove the carboxyl group in a decarboxylation reaction. In either case, the organic molecule that's left behind can often enter the Krebs cycle or one of the other stages of glucose catabolism as a reaction intermediate. Like other biochemical reactions, we can use the products of protein catabolism in the laboratory for the identification of unknown organisms. And there are several different tests that look at amino acid deamination and amino acid decarboxylation. Although I won't go into detail, nucleic acids and polysaccharides can also be catabolized by microorganisms. Their breakdown can feed into the glucose catabolism pathways as well. This is why we focus on glucose catabolism, because it is what we call the central metabolic pathway. While we have focused almost entirely on using organic molecules for energy, it is also important to think about how food can be used as a carbon source to build biological molecules. Remember, most chemoheterotrophs can use glucose as both an energy source and a carbon source. Many of the intermediates that are made during glucose metabolism can also be used to make the building blocks of our biological molecules. How the cells utilize glucose depends on their carbon and energy needs at any given moment. Pathways that can be used for both catabolic and anabolic purposes are known as amphibolic pathways. So you can see these arrows on my screen, they go in both directions because depending on what the cell needs, it can either build proteins or break them down, right? And it can use the intermediates of the glycolytic pathway and the Krebs cycle to do so. All right, so this is the end of our series on metabolism. Hopefully you have a basic understanding of the biochemical processes used by cells to grow and thrive. You should also be able to see how we can use this information to characterize and identify microorganisms in the laboratory.